Okay, class. So I welcome you all to another lecture. And in today's class, we would be looking on toward that what are those factors that would be affecting the enzymatic activity. Earlier, we know is that in our body there is existence of these biological catalysts, and these biological catalysts we were entitling them to be as called as the enzymes we say, and we were clear that within the enzymes there lie this site, and this site is the active site. And within the active site, the substrate molecule would come, it would bind here, and as a result of that, a complex would be formed, and that would be the enzyme substrate complex. So today we will look at what are those factors that would contribute to this more formation of the enzyme substrate complexes. Because if more enzyme substrate complexes would form, then ultimately the rate of the chemical reaction would be faster, and that would benefit multiple chemical reactions that are important for the survival of the living organisms. For your level, you need to be knowing is that there are a total of four of the factors that would be affecting the enzymatic activity, and among them, the first factor that we have with us, this is the temperature. After the temperature, we have the pH. After the pH, there would be coming up of that how much amount of the substrate we have with us. And then there would be that how much of the enzyme amount we have with us or the enzyme concentration we have with us. Now, as far as the word concentration is concerned, you just need to know as that if I would take a beaker, and in that beaker, if I take a volume of something as 1 liter or 1 dm cube, so how much amount of the enzyme or how much amount of the substrate would be available in that 1 dm cube. So this would be the concentration. So these four factors, they decide the enzymatic activity. But we must be very much clear about that how come the temperature and the pH graphs they look like and how they would have an impact on the enzymatic activity. But before I take you to be understanding these four factors that we have got with us, now there is one thing that we must be aware of, that here you see I have got this enzyme, and for this enzyme the shape of the active site it looks like this, and we say that this is the three-dimensional shape of the enzyme's active site, and this shape of the active site would be different for every particular enzyme that we come across. Like if I were to show you some other enzyme, and this enzyme would look like this, and in the enzyme the shape of the active site would go like this. So you would not be able to justify these factors until or unless you have an idea of that what actually lies within the enzyme molecule, which would be maintaining this three-dimensional shape or three-dimensional configuration of the active site. Because if we will not maintain the three-dimensional configuration of the active site, then ultimately the enzyme molecule would not be able to be working on some substrate. Now, earlier I have said that these enzymes they are the proteins in nature. Now, if they are the protein in nature, then we know that here we would have the amino acids, and these amino acids they would be linked together with the help of the peptide bond. And as a result of that, we are making these chains out of it, and these chains they are the polypeptide chains. So the situation here is that here I have got this first polypeptide chain, here I have got this second polypeptide chain, and in between the amino acids, there would be existence of some attractive forces. And these attractive forces, as far as they would remain, so the enzyme would be maintaining the shape of the active site, and when it would maintain the shape of the active site, then it would be working on toward the substrate which have to be catalyzed. If I were to show you a detailed diagram of what I was speaking at the moment, so you see that here I have got these polypeptide chains within the enzyme molecules, and here we have the amino acids within the polypeptide chains, and in between the amino acid there would be existence of some attractive forces. At higher level, you would learn that these attractive forces they include the hydrogen bonds, they are the ionic bonds, they are the disulfide bridges, they are the weak hydrophobic interaction. But for your level, you just have to remember is that as far as in between the amino acids these attractive forces would lie, then the enzyme would maintain the shape of its active site, and that would be allowing an enzyme to be working on the substrate. So we would see that if we try to be changing the temperature and pH, then something would happen to these attractive forces between the amino acid, and in some cases the enzymatic reaction would come to an end, or this is gonna cease. So now before I go further, now here you see that we have got this graph to be understanding that how come the temperature would have an impact on the enzymatic reaction. Okay. Now, since we have to see the graph, so for the graphs, what you remember is that anything that we have on toward the x-axis, so this would be my variable that I would change in an investigation. Because if I were to see the effect of the temperature on the enzymatic activity, then I would take the enzyme, I would take the substrate, and then I would be keeping that solution of the enzyme and substrate at different temperature to see that how come the enzyme is now working on the substrate. So my temperature would come on toward the x-axis, the horizontal axis that I have, and here we are labeling it up in terms of temperature in centigrades. And on toward the y-axis here would come the rate of the enzymatic activity. Now, if you see for yourself, like as the temperature is increasing, like when we are moving from the zero value to this side, so onto this side, the temperature is increasing. And if you look on toward the graph, so as the temperature is increasing, so the rate of the enzymatic activity is also increasing. And here you see at this particular point, the enzymatic activity is reaching the maximum rate. Why I'm saying this is reaching the maximum rate? Because for our curve that we have got, so this is reaching the highest value at this level. So you have to remember is that temperature at which the enzyme would exhibit the maximum catalytic activity. So such temperature is what we refer to it, or we call this as an optimum temperature, we say. Now, you should always be in the position to know that how every part of the curve that we have here, you would explain it to your examiner. Okay, now you see, you would say that as the temperature is increased from 0 to this value that we have got for most of the enzymes that are working in the human body, so their optimum temperature where they exhibit maximum catalytic activity, so this is 38 degrees centigrade. So as the temperature is increased, you see that the enzyme and the substrate kinetic energy would increase. And when the speed of the enzyme and the speed of the substrate would increase, so this means that they would move more randomly. And when they would start moving more randomly, so there is a greater chance that the substrate would fit into the enzyme's active site and enzyme substrate complexes would be formed. And ultimately, when we would come to this point where we have this optimum temperature and the maximum enzymatic activity, so at this point, maximum enzyme substrate complexes would be formed, and that is why the enzyme is exhibiting the maximum of the catalytic activity here. But if you see it for yourself, if I am increasing the temperature from 38 onward, so you see that the curve is falling downward. So when the curve is falling downward, so this does not mean that the enzyme has completely stopped acting on toward the substrate, but you see that the frequency at which the enzyme was binding with the substrate, so this is decreasing. The reason here is that when you would increase the temperature beyond 38 degrees centigrade in case of the human enzymes, then as a result of that, in between the amino acids that we have and that are basically determining the three-dimensional configuration of the enzyme's active site, so the attractive forces between the amino acids, they would start to break down. And when these attractive forces between the amino acid, they would start to break down. So the enzyme would start to lose the shape of its active site. And this is less likely for the substrate, like substrate still would bind with the enzyme's active site, but their binding would be comparatively less as when we have the range of temperature in between 38 degrees centigrade or between beyond 30 degree and 38 degrees centigrade. Okay. And when you see that at this point where the curve has touched nearly the x-axis, so here what we say is that all those attractive forces that were lying in between the amino acid and were determining the three-dimensional configuration of the enzyme's active site, so they would be completely broken down. And when they would completely broken down, so as a result of that, we use a word that the enzyme would be completely denatured. Now you see that when the enzyme has got completely denatured, then none of the substrate would bind with the enzyme's active site, and ultimately the enzymatic reaction would come to an end. Okay, so what you have to see is that beyond this, the enzyme is still catalyzing the reaction. As far as the curve is not touching the x-axis, there would be some of the chemical reaction happening, but that chemical reaction is not as much frequent as we have got on this part of the graph. And when you would be saying this in terms of this part, so we will say that the attractive forces between the amino acids, they have started to broken down. Enzyme has started to lose the shape of its active site until here when the curve has touched the x-axis. So here all the attractive forces in the amino acid that were determining the three-dimensional shape of the enzyme's active site, they would be completely broken down. The enzymes become completely denatured, and when this becomes completely denatured, then the enzymatic reaction would stop here. So I have written like each and every explanation for the part of the curve that I have got. So here let us just try to see that what you need to write in terms of explaining the graph that we see in context of the area A that we have. So for the area A, you would say that as the temperature increases, so the enzymatic activity increases. And the reason is that with an increase in the temperature, the kinetic energy of the substrate and the enzyme they both would increase, and the collisions between the substrate and enzyme they would increase. And as a result of that, more enzyme substrate complexes would begin to form. And ultimately, when we have reached here at B, so we have got the maximum catalytic activity. And if you have to explain in terms of the point B, like the description that you have to know is that at B, the enzyme molecule they attain their maximum catalytic activity. And here, most of the enzymes active site, like many of the enzymes that we have, so their active site, they would be occupied by the substrate. So here we have attained the maximum catalytic activity. And how you see that that point where the enzyme has attained the maximum catalytic activity, so this is the enzyme's optimum temperature. And this varies from individual to individual, from species to species. If I were to give you an example of us, the human beings, so in our body, the optimum temperature is 38 degrees centigrade. But for the bacteria, those who live in the hot springs, so their optimum temperature would not be 38, but rather than it would be much more greater than 38 degrees centigrade. Now, for the part of the graph that is labeled as C, so you see as that when the temperature is increased beyond the optimum temperature, the enzymatic activity this begins to decrease. Why this begins to decrease? Because for the enzyme's active site that was determined by the amino acids, and among the polypeptide chains you see that we have got these attractive forces. So these attractive forces between the amino acids for an enzyme molecule, they would start to break. They are not completely broken now. The word is start to break. An enzyme would start to lose the shape of its active site, and less substrate will bind with the enzyme's active site. So that is why the curve is going downward. And when we come at this point D, where the curve has touched the x-axis, so you would say that here the enzyme has become completely denatured. The enzymatic reaction would stop, and the reason is that the enzyme has got completely denatured, like it will lose the shape of its active site. Like initially, the enzyme whose active site was looking like this, so this is completely deformed, and this is completely deformed, so no substrate is gonna bind with the enzyme's active site. Now, the other factor that we have to be very much clear about is that how come the pH would be affecting the enzyme's activity. Now, if I were to give you an idea of the word pH, so pH simply means the part that we've got for the hydrogen ions. Like overall in our body, we have got total of three types of the pH in case of the human body. Like we will have this acidic pH, then we will have the neutral pH, and then we will have the alkaline pH. Okay, now the wherever the word acidity is there, so this would be that how much of the H positive ions I have with me. So if I would have more of the H positive ions, so the pH value would be less. Okay, and that lower getting the value of the pH getting lower. So this is like how much amount of the H positive ions we have with us. So wherever the H positive ions would be more, the pH would be less. It would have lower values like 1, 2, 3 in that range. And whenever we have the neutral pH, so we say that at that pH, the number of H positive ions that we have and the number of OH negative ions that we have, so they would be equal to one another. So overall, the pH would be neutral, and the neutral pH actually has a value of 7. The third pH that we have to need to have an idea of, so this is the alkaline pH, and this alkaline pH is due to OH negative ions. Like more OH negative ions we will have, so greater would be the value of the pH, and that would be beyond 7. You may have learned in your chemistry that the pH is basically expressed or is used to is determined with the help of the scale that we have, and the pH scale has a value ranging from 0 and max it comes till 14. From 0 to 7, we have the acidic pH. From 7 to 14, we have the alkaline pH. And as we are moving toward the 14th value, so the number of OH negative ions is increasing. And when I am going back from 7 and going toward like the value of 1 or 0, so here the H positive ion concentration is increasing. And this would be the acidic pH. Okay, the lower the value of the pH, more acidity there is. The higher the value of the pH scale, more alkalinity there is. And when the number of H positive and OH negative ions that we have with us, so when this is equivalent, so this is the neutral pH, and in the human body, most of the enzymes they prefer to work at a neutral pH. The stomach enzymes that we have in our body, they would work at the acidic pH. And the enzymes that we have got in the small intestines of our alimentary canal, so they would be working in the alkaline pH. Now again, the situation is like if I were to take the enzyme from the stomach that prefer the acidic pH, and if I introduce it to the alkaline pH that we have in the small intestines, the enzyme would again denature. And again, vice versa, if I were to introduce the intestinal enzymes that prefer to work in the alkaline pH, and if I would introduce them into the acidic pH that we have in the stomach, so likewise they would also denature. So along with the temperature, this pH again, you see that this has got an impact on the extent to which the enzymes can be working on their substrate. Like if I were to tell you about this graph, so this graph is basically drawn for the enzyme, and that enzyme is working in the duodenum of the small intestine. Now the duodenum of the small intestine basically has the alkaline pH. So what we expect is that the enzyme would have maximum catalytic activity beyond 7. So you see that at this point where the curve has touched the x-axis, so here you see this is high acidic environment, or here we have got a lower pH value. So that lower pH would again break down the attractive forces that we have in between the amino acids, and the enzyme would be completely denatured. But as the pH is increased from 2, and ultimately we are going in the alkaline range, and here you see at the 8th pH for the duodenum of the small intestine, the enzyme again is attaining maximum catalytic activity. So here you have to note at this point where we have got the peak of the curve, so this would be the optimum pH, and this optimum pH means that pH where the enzyme would exhibit maximum catalytic activity. And for every particular enzyme, the optimum pH value would be different. If this was a stomach enzyme, then the curve would have a peak at a pH of 2. If there was a neutral enzyme, so the curve would have a peak at the pH of 7. Okay, and likewise, if I have got this small intestine enzymes coming that is working in the duodenum of the small intestine, so here the optimum pH value is 8, and beyond 8 you see that at this point again the enzyme would start to denature. This would be binding with some of the substrates, but the binding with the substrate would be becoming less likely as we see an increase in the pH. And when we have reached toward a very higher alkaline pH, here you see at 13 pH, so here you see that all the attractive forces between the amino acids they would be broken down, and at this point the enzyme would become completely denatured. So this was all in terms of the factor that would impact enzymatic activity. For your level, you just have to be clear about it, how the pH impacts the enzymatic activity and how the temperature would have an impact on the enzymatic activity. But you must know that there are four of the factors that can affect enzymatic activity: the temperature, the pH, the amount of the substrate, and the amount of the enzyme that we have got.